to join us. All right. So just some announcements kind of just about the rest of the semester. So hopefully you read my email yesterday. So instead of having sort of like a high stakes unit 3 and final exam, we're still going to have these in sort of like an exam format, but it'll be through Connect. So there's a couple benefits to doing this. You've seen a lot of these questions before, so it'll be reviewing a lot of the information that you've acquired through LearnSmart and Connect, similar things there. I do restrict those questions to what we have learned in lecture. So the questions won't necessarily be from the LearnSmart, they'll be from kind of Connect homeworks, but also like the study guides from the PowerPoints will still be extremely beneficial for studying. So the questions will be directly related to stuff we covered in lecture. The LearnSmarts are meant to be a little bit more broad than what we focus on in lecture, but both the Unit 3 exam and the final will focus on what we covered specifically in lecture. So Unit 3 will be genetics and metabolism, and that will go live on Sunday, and you'll have all of next week to do that. And then the final exam will go live next Sunday, and you'll have until Thursday to do that. So the Unit 3 is not going to be timed. You'll have 2 attempts to do it, and your best score will be recorded. And then the final exam, though, is going to be timed. You'll have about 2 minutes per question, sort of like with the last exam. But again, it's through Connect. And the reason I'm doing it through Connect instead of like on Canvas is because if I do it through Connect, that way you can kind of like use your resources, right? There's no lockdown browser. So you should still be able to access like your textbook and your notes and stuff like that electronically, as opposed to difficulties with the Respondus. So note that I said that, right? So you can use those resources if you would like. Again, note you only have 2 minutes per question. So make sure you know it and you're prepared, but that's all available. So you'll have about a week to do that as well. So that'll all go live, and I'll continue to send out announcements. One really important announcement that I wanted to mention is that if you are not caught up in lab, so I think a lot of you, and I need to double check my lab sections to see, but remember that we have the rule that missing 2 labs will make you fail lab. So I'm not 100% sure how the department is handling that rule. I'm going to reach out today. Obviously, I want to make sure that everybody has the opportunity to complete the labs. So with those Labster labs, if you are behind on finishing those, especially if you're behind on like 2 of those, prioritize that. Okay, so we probably won't stick to the missing 2 labs and fail a lab rule, but you do need to make sure. I think that what we'll do is we'll end up giving like an extension for when those are due. So if you haven't done them already, reach out to your lab professor. If that's me, reach out to me, or if it's Shanna or Rashad, reach out to Shanna or Rashad and just say, I need a little more time for whichever Labster stimulation. But the really important thing here is that we could hear from the department that we have to stick to that rule where missing 2 labs equals failure of the course. So in that case, what you definitely need to make sure you do is just get those labs in. Again, flexible deadlines, we'll send out those deadlines, but just do it. That's the important thing. But again, reach out to your lab professor. We're definitely willing to work with you. We just want you to do it. All right. So, yeah. So get those labs done. Okay. So I think that's all of the announcements today. Yeah. Except for I put on eyeliner. So you're welcome. Feeling fancy today. Okay. So let's go ahead and get started. So share my screen. I am recording. Check. make sure. Okay, so let's look here. And I need this. Come on, there we go. I want to activate my chat. Great, we got the chat going. We got the lecture going. We're recording. Stuff is good. Okay, so just kind of a reminder where we left off last Thursday. We were talking about gene regulation. So hopefully you got my message that I did go through and update the PowerPoint just a little bit. My hope is that it clarified a little bit of the confusion that I saw as I was going through the discussion board. I also went through the discussion board as well, so if you have any questions, make sure you read through the discussion board, because a lot of those questions were really good questions, and I tried my best to answer a lot of those in the discussion board as well. But just a reminder that when we have the process of gene expression or transcription and translation or central dogma or protein synthesis, all of which mean the same thing, which is the process of turning DNA into proteins, that occurs in two major steps: transcription, where we turn DNA into RNA, which occurs in the nucleus. Reminder that when we do that, the first step is that we turn DNA into that pre-mRNA, which is the mRNA that's an exact copy of that DNA. So just a reminder that that pre-mRNA contains the introns and exons. So what we have to do after we have that pre-mRNA is we have to process it. So whenever we process it, we add a cap, we add a tail, and we splice out the introns. So we're left only with the exons. And those exons are what basically make into codons, and then codons code for amino acids. So what that means is that the exons are directly responsible for the sequence of amino acids, therefore our primary protein structure. And since structure equals function, that exon sequence is absolutely vital to the protein we're going to end up with. So that's our first step, transcription, taking DNA, turning it to pre-mRNA, processing it to get our mRNA. Then that mRNA has to leave the nucleus. Once it gets into the cytosol, it has to associate with a ribosome, where it will be translated codon by codon into amino acids, which is going to be our protein. So we understand that process, and what we have been talking about is basically how can we regulate that process. And when we talk about regulation, what we're talking about is how can we basically say, yes, we want this gene transcribed, or no, we don't want this gene transcribed. So we talked about one of the most important steps being number one, which is transcriptional control. Oh, well, we also talked about the fact that DNA methylation, methylation and acetylation of the histones is sort of like before transcriptional control, like how available is your DNA for transcription. We talked how methylation tightly winds that DNA, so it's inaccessible, transcriptionally silent or not active. Or if we acetylate it, it opens it up so that it can be active. It can be transcribed, and that would be Ubiquitination. All right, so we talked about epigenetic changes, which is another important step for methylation. Epigenetics is when individual nucleotides in the gene sequence are methylated. So I had a couple of really good questions about understanding how epigenetic change causes a change in the phenotype without altering the genotype. And so to kind of answer this, I just want to remind you that like a genotype is the sequence of nucleotides in a gene. So for example, ATTCCG. Whenever we have an epigenetic change, all that we're doing is we're adding, let's see if I can do this. How can I be clever about this? All that we're doing is we are taking a methyl group and methylating it, or methylating a nucleotide. I'm probably going, well, whatever. You guys get the point. That's a methyl group. So all we're doing here is adding a methyl group to one of the nucleotides. Typically, this is going to be a cytosine. So notice that our nucleotide sequence, so the actually genetic sequence, our genotype, was not altered by this. But this little chemical change, what that could do is that, or what it could mean, is that when we go through transcription, right, so let's just say this is going through transcription, what is actually transcribed would be, so remember we do complementary base pairing to get an RNA sequence, so A, U, T, A, C, G, and then when we get to this methylated cytosine, what could happen is two things. One, it could stop transcription, so RNA polymerase doesn't recognize it and kind of disassociates, or it could just like skip over it, so we would have G. So you'll notice that this basically has a very similar effect as what we'll talk about later, which is an actual genetic mutation. But the difference here is that the actual genotype, right, this sequence hasn't changed. So the genotype hasn't changed. But if we change our mRNA, we're going to change the amino acid sequence, and therefore our phenotype will change. So today, we're going to move into genetic mutations where we have a change in genotype, and obviously this may or may not result in a change in phenotype. So the big difference between epigenetic changes and genetic changes is either one may or may not have a result, but epigenetic changes cause a change in phenotype without changing the sequence of DNA, whereas genetic changes may or may not cause a change in phenotype, but they definitely have an effect, as we'll talk about on genotype. So again, we'll elaborate more on that later, but I just wanted to kind of clarify that with epigenetics, you're not changing the sequence of nucleotides in the DNA. And that's what we mean by it changes it doesn't change the genotype, but it does change the phenotype. Right. So we talked about genetic regulation there. We also talked about how kind of the most important level of gene regulation is this first step, which is using transcription factors to basically determine will this gene be transcribed or not. So remember that for any gene to be transcribed, we use transcription factors. And those transcription factors include proteins that bind to the TATA box to form the pre-initiation complex at the core promoter, but they also involve this process where activators bind to an upstream segment of DNA called enhancers, causing those activators to bind to the pre-initiation complex, basically starting the process of transcription. So remember, I linked that video, and this video does a really pretty good job as well, kind of explaining that process. So those are transcription factors, level 1 of how we can say yes or no to gene regulation or to gene transcription. We can also regulate how kind of the next step, how RNA is processed. This is one of those really cool things we talked about. This was also a common question in the discussion board, and I went through and tried to answer some of those questions. Somebody else did like a really, really good visual explanation of this as well, using kind of like colored sequences. I can't remember who did that, but thank you, that was beautiful. But basically what we talked about with RNA splicing is that in any sequence of DNA, you have a combination of exons. So for example, here we have 5 exons. We go through transcription and we get RNA with those same 5 exons, as well as the introns, which are shown in pink. And via splicing, what we have the capability to do is basically say, like, this segment of RNA will only contain 4 out of those 5 exons. So let's say randomly 1, 2, 3, and 5. And then we can also splice it to where a whole different piece of mRNA has a different combination of 4 of those exons. But remember, since protein structure is dependent on the exons present in the mRNA, if this mRNA here on the left only has exons 1, 2, 3, and 5, that's going to code for like protein number 1. We can add that in here. Oops. That sequence of exons would code for like 1 protein. And that would mean that this other sequence, because it's a different sequence of exons, there's different exons present, it will lead to a completely different protein. So that's why with any sequence of DNA, like this is a gene, the more exons that are present in that gene, the more sequences we can create. And the more sequences we create, the more possibilities of proteins we can make. So that's what we were talking here, is that in the human genome, we actually only have around like 21,000 genes or so. But with that limited amount of genes, because every gene has lots of combinations of exons, we can make way over 100,000 proteins. And same with Drosophila, which is a fruit fly. That's just like one example of an organism that takes this to the extreme. Drosophila has one of its genes that is alternatively spliced so much that it results in the production of like 17,500 proteins, which is crazy. So that's just an example. So that's another way that we can kind of regulate transcription, but instead of regulating it where we're taking DNA and turning it into RNA, we're regulating it at the RNA processing, so pre-RNA to RNA. And then we can also regulate this process as the RNA kind of leaves the nucleus, basically just how accessible it is and like its activation. So we talked about how some RNA can't be translated without certain signals, and that how some mRNA hangs around for a long time, whereas others are relatively short-lived. So the importance of this, like why an mRNA wouldn't hang out a long time, comes down to the fact that basically once we get to the point of having mRNA created, it's very likely that that protein will be made. So the reason we only want to kind of make mRNA as we need it is because we really want to be able to regulate or determine our phenotype based on the circumstances, right? So we kind of talked about this a little bit, where this is very environmental. So for example, I don't want to transcribe all of the genes I need to run a marathon right now because I'm going to be sitting in front of my computer all day. So I would be wasting a ton of energy making all of the proteins necessary to run a marathon, and I would be just ready to run a marathon but sitting here, which would be very distracting, right? So I only want to make the genes necessary for sitting in front of my computer all day today. If I was going to run a marathon, you would be preparing by drinking water, hopefully preparing by drinking water and taking in carbs and sort of like, jogging at the start site, warming up your body. And that would start turning on those genes that prepare you to run a marathon. So again, once you make mRNA, it's pretty much committed to a phenotype. So one of the reasons we don't make mRNA that lasts a long time is because we basically only want to make mRNA right when we need it, or right when we need a certain protein. So that allows us to kind of switch conditionally based on what we need at a given minute. It's pretty amazing. We also talked a little bit about how we can do kind of moving away from transcriptional control and how we're controlling the mRNA. We talked about, like, let's say we have mRNA, but we just like don't want it to be made into a protein again. So we committed to making that mRNA. How can we stop this now? And we talked about the fact that we have some small non-coding RNAs. So remember that non-coding means that these are not going to be committed to making a protein. And instead, pretty much what they function to do, which you're seeing them in yellow, and our mRNA is in pink. Remember that mRNA has to be single-stranded to be read by a ribosome. So what these non-coding RNAs do is they basically bind complementarily to the mRNA. And once the mRNA is double-stranded, it can't be read. So it's basically a way to say, oh, we have this mRNA, we don't want to use it anymore. Use one of those small non-coding RNAs to just like turn it off. So we might call that RNA interference. Sometimes it's called like interfering RNA. Those have minute differences as far as function, but we're not going to go into details with those. All right. And then I wanted to finish up with this portion, regulation, today, talking about like, okay, so we got through transcription, we got through processing, we got out of the nucleus, we're active, we didn't get stopped by a small non-coding RNA, and we were translated into a protein. We can even still basically regulate whether that protein will be active or not. So there's two primary ways that protein function is regulated. So basically, can we use this protein or not? And those two methods are via cleavage, which is activation, and then degradation via ubiquitination. So let's talk about those. So the first is called cleavage. So anytime you hear the term cleavage in biology, basically what it means is just kind of like cutting something. So because of the fact that, sort of like for the same reason that the majority of the genes in our body are actually turned off, and we have to turn them on with transcription factors and activators, right? The majority of our genes are off. Similarly, the majority of the proteins we end up making are off. We have to basically say, oh, we made this protein. Do we really need it? Yes. So how do we get that yes to say like, yes, we need this. Yes, it should be activated. And the answer is via some types of protein modification. So there's actually a lot of different types of protein modification. It can be chemical modification, so very similar to like methylation, or phosphorylation, or acetylation, or glycosylation. There are lots of different chemical modifications of proteins that have to take place post-translationally. But we're just going to talk about this one today, which is cleavage. So this is talking about, again, just like kind of like cutting up a protein after it's been made. There are two reasons that you might do this. One of the first is exemplified in the picture at the bottom of the slide. And it's when you may have like an inactive protein or polypeptide, and in order for it to be activated or turned on, it needs to be cut. So that is actually exemplified by a pretty common protein. For example, if you've ever heard of insulin, insulin is the protein necessary. You have to produce insulin in order for glucose or sugar, which will create energy, in order for glucose to be allowed into cells. So insulin basically allows glucose to enter your cells. If you don't have insulin, you can't get glucose into your cells, and therefore you have reduced capacity to generate energy effectively. So here, insulin is kind of like a dose response, right? So we know that we need insulin pretty much all the time because we are taking in sugar and need to turn it into energy all the time. But you don't want to have too much glucose moving into your cells at a time. So we know that we're going to need to move glucose in. We just don't know kind of how much. So insulin is one of those proteins that we actually make a lot of. Insulin is made pretty much all the time. We do have transcription factors for it, but we don't really stop the process. We're just saying like, yeah, we're probably going to need this protein. Let's make a lot of it. However, we don't want it to be activated until we need it, right? So for example, I don't need a ton of insulin to move glucose into my cells right now, but I will need it if I eat a candy bar, taking in a ton of sugar. I need to start moving that sugar into my cells for energy, right? So we only really need insulin when we have a lot of sugar present that we need to move into our cells. So insulin is made in an inactive form. So you can kind of see this purple would be a polypeptide, and it is activated by having what are called cleavage sites, where it's basically just like cut. And you can see once it's cut, that would be insulin activated. So one of the things that happens when you take in, like you eat a candy bar or something, is your body says, ooh, I need insulin. And that is an activator for that cleavage. So you're cleaving insulin so that it can be activated and it can pull that sugar into your cells. So that's one example of why cleavage is important, right? If you have a protein made and it's made in an inactive form and you want to activate it. Another reason that cleavage is important is what you can sometimes do is very similar to like how mRNA, pre-mRNA splicing can result in lots of different, one pre-mRNA resulting in several RNAs and therefore several proteins. We can also have one initial, what we call polyprotein. So for example, it's like a really long strand of a lot of proteins built together. And then what we can do is we can cleave it. And then instead of having one poly meaning multiple, instead of having one polyprotein, what you would do post cleavage is that would kind of cut it to where you have lots of different proteins. So an example of this would be, as we will learn, like cellular respiration is a metabolic process. Metabolic processes require tons and tons and tons of machinery. And that machinery is in the form of primarily enzymes, and enzymes are proteins. But for example, like step one, which is glycolysis, where you take, literally you take a 6-carbon sugar and break it in half. Way more complicated than that, but that's what you do, right? Glycolysis. Glyc means sugar and lysis means splitting, so you're splitting it in half. That requires several enzymes. And if you're going to have a sugar and break it in half, you want to make sure you have all of the machinery necessary to do that. So one way you can ensure that all of the machinery necessary for that one event is present is you can create all of that machinery as like one giant protein, polyprotein, cleave it into multiple active proteins. So you may create one polyprotein composed of like 5 proteins bound together, and then you cleave it, and then you have 5 individual proteins. So again, it's just a way to kind of conserve energy, doing this all at once, but make sure that you have all the machinery necessary for like one process at the same time. So those are 2 kind of reasons why we might use cleavage. I do want to mention again that this is not the only form of protein modification. It's not even the most common form of protein modification, but it is a very important one. So that's how we can activate proteins. So basically we make a protein in an inactive state and then we activate it. However, just like we talked about with mRNA, we don't really want proteins or mRNA to hang out around a long time because we want to be able to change our phenotype as necessary, right? I only need a ton of insulin if I'm going to be eating something, because only then do I need to be importing that into my cells. All other times I don't need that much insulin. So I need to make sure that, right, I have the machinery when I need it, and I'm not wasting the energy to make it when I don't need it. Same with proteins. You know, if we get to the point where we have proteins, then we have a phenotype. So how do we kind of like erase that phenotype and start over, right? How do I go from sitting on the stool to creating a new phenotype where I'm ready to run a marathon? It's a very extreme example because it's not going to happen. Maybe someday. But you get my drift, right? How do I go from fasting, right? So overnight, not eating for so long, to being prepared to eat breakfast? How does my body prepare for that switch, not eating to eating? So aside from changing the genes, we also have to kind of get rid of all of the proteins we have and create new ones. As well as that, proteins, when they're made, are like any other chemical in that they kind of like age, and it's better when they're fresh. Because what happens is, as a protein hangs out, it can accumulate kind of damage and mutations and all this stuff. And basically we want to make sure that all the proteins we're using are fresh and good, and basically just the most functional form of that protein possible. So what we do with proteins is, on the left here, in this little blue protein, let's say that would be like a freshly made protein. As a protein hangs out after it's been made, it accumulates a molecule. So this is sort of like that methyl group just being attached to a nucleotide. Instead, we have this little molecule kind of being attached to this protein. You can see this protein over here has a couple of them, but you see that there's one right there. You have this little molecule, and it's called ubiquitin, right? So ubiquitin, you can kind of think of ubiquitin as sort of like a wrinkle, right? So I've been here for a while on this earth, and so therefore I'm getting older, and therefore I'm accumulating wrinkles, right? So wrinkles tend to show our age. The presence of ubiquitin tends to show the age of a protein, because proteins accumulate ubiquitin over time. So for example, again, you have this protein that was just made. Over time, it's accumulating ubiquitin. The process of accumulating ubiquitin is called ubiquitination. And the enzyme that does this, that kind of adds ubiquitin, is called ubiquitin ligase. So remember, the last time we heard about an enzyme called ligase was during replication, where we're connecting or gluing those Okazaki fragments together. In this case, ubiquitin ligase is kind of like gluing ubiquitin to a protein. So it's kind of gluing it. Is that the same thing as the vitamin ubiquinol? I don't know. Let me look that up. I'm going to write that down. I don't know if I want to admit this or not, but I've never heard of that vitamin. So let me copy that question. Oh my gosh. I need a new keyboard. Okay. Let me look that up because I'm not really sure. I will get back to you about that. Good question. All right. So the enzyme ubiquitin ligase, what that does is it adds ubiquitin to those proteins as we go. So again, you can kind of see what happens is if you have fresh protein, we start accumulating those ubiquitins. And essentially what happens is, after a certain amount of time, if a protein has accumulated enough ubiquitin, the body's kind of like, it's run its course, right? It's been around long enough, right? So when I'm 135, I will probably have accumulated enough wrinkles to pass. We'll see. I don't want to live that long. Okay, but right? That's what happens is as you accumulate those, as a protein accumulates those ubiquitins, your cell can recognize this protein's been around for quite a while, it's probably time to replace. So what happens after it has been, or after it accumulates that ubiquitin, is it basically is targeted for degradation or breakdown. And the complex that does this, the organelle that does this, is called the proteasome. And what the proteasome does is it has a bunch of enzymes, a lot of terms on this slide, a lot of enzymes. Remember, ASE tells you it's an enzyme, and this tells you exactly what it does. Proteases break down proteins. So that's exactly what happens. And you can see what it does is it breaks down proteins into individual peptides, can break it down into individual amino acids. So this is one of the ways that what we can do is if you eat like a protein source, you actually have proteases in your stomach, and those proteases break down proteins, like the protein you get from meat or quinoa, and it breaks down that protein individual amino acids, which can then be used for translation to build new proteins. So proteasomes and proteases are some very, very important enzymes and organelles in the body that help break down proteins. They do that for digestion, but they also do that via the process of, or subsequent to the process of ubiquitination, basically just to make sure that we're replenishing proteins kind of as we go. So I'm actually really curious about ubiquinol. Let's look that up. Protect the body's, oh, let me look more into it. But this says that ubiquinol, I mean, obviously very good research here, just via straight Google search. But this says that it contains a form of CoQ. So we will talk more about CoQ a little later when we talk about metabolism. But from what I understand, let me look more into that. So I don't think that it is the same enzyme, but sometimes they give those CoQs kind of a weird name. But let me look into that more closely. So I guess like based on preliminary Google research, it is not the same as the enzyme ubiquinol, but let me see what it does. And I had another question. Does ubiquitin get deposited equally on all proteins, or does the ubiquitin ligase differentiate young and old proteins? That's an excellent question. As for how it is deposited, there are a lot of different ways that it's deposited. So we're not going to go too much into detail, but there's two primary things that can happen. So number one, basically is kind of what we talked about here, is that freshly made proteins will accumulate ubiquitin over their lifespan. So yes, freshly made proteins are ubiquitinated, but that doesn't mean that they're automatically targeted for destruction, because notice, like, even if we have one ubiquitin attached to this protein, you have to accumulate quite a bit, right? So I'm not going to pass after I get my very first wrinkle, right? So just one wrinkle is not a signal of, you know, you've run your course. I'm going to pass once I've accumulated lots and lots of wrinkles. That's a correlation, not causation. Very important. But here, the thing is like, yes, you can get young proteins ubiquitinated, but that doesn't mean they're going to be targeted for destruction, right? So only ones that have accumulated quite a bit of ubiquitin will be targeted for destruction. On the flip side, ubiquitination can also be used as a really good way to kind of like target proteins for destruction. Let's say if they're like misfolded or non-functional. So your body or your cells can basically, like, what happens is as they move through the endomembrane system, which remember is like from the nucleus to the rough endoplasmic reticulum to the Golgi through vesicles to the plasma membrane. One of the things that happens in the Golgi is that they are modified. So it's in the Golgi that they would be cleaved or chemically modified by phosphorylation or acetylation or glycosylation, whatever. It's also in the Golgi where they're basically just like checked, right? Is this folded correctly or is it folded incorrectly? Was there a genetic mutation or an epigenetic mutation? Like, is this protein right? And one of the things that can happen is if the Golgi detects that the protein is misfolded or is in the incorrect form or is misfunctional somehow, one of the things that it can do to basically target for destruction is ubiquitinate it. So this ubiquitination does take place over the lifespan of a protein to kind of target it for destruction eventually, but it can also be used as a way to kind of signal a bad protein so that it is destroyed as well. Excellent questions. Good, good, good. All right. So, cool. Let's move on. I do want to point out just one thing, though. This is one of my favorite slides. That doesn't necessarily mean it's one of the most important, but I think this slide is crazy. What this pie chart is showing you is basically all of the DNA, right? So this entire pie would be all of your human genome and are all of the nucleotides in the human genome. So the kind of crazy thing about this is that you can see that in your genome, 19% of all of your DNA is in the form of introns, which is, remember, the non-exonic or non-coding DNA. 15% of your DNA is unique non-coding DNA. So we're not going to talk too much about that, but that's very similar to these introns. We're not going to go into it. 15% is actually just like repetitive DNA, which is sort of weird. Transposable elements, which are possible historical evidence of viral infections, basically. And then 44% is some more repetitive DNA. So essentially what that means is that whatever that is, 19 plus 15 plus 15 plus 44% of this, all of this, it is non-coding DNA. And then you look at this kind of like medium purple color. That medium purple color is regulatory sequences. So that's DNA that's directly involved in regulating the transcription or translation of the genes. And then if you look at the dark purple area of this pie chart, the dark purple area of this pie chart is referring to your coding DNA. So the amount of DNA that's directly dedicated to creating proteins, genes that code for proteins. 1.5% of your entire genome is composed of exons that code for proteins. The rest of it is regulatory, so 5% of it is regulatory. And that means the vast majority of it, what is that, like 93.5 or 94.5 of it, is non-coding, repetitive exon or introns, which, remember, have a regulatory function. So that is a lot of DNA where it's not directly coding for proteins. So way more, around 4 times as much DNA is dedicated to regulating that 1.5% of exonic DNA, which to me is just literally insane. So I like showing this pie chart just to emphasize the fact that we talk so much about protein, making proteins, right? DNA makes proteins, DNA makes proteins, but really the quantity of DNA that you have that directly codes for proteins is so small. It's extremely important, but the vast majority of your DNA actually has other functions. Unfortunately, we're not going into hyper depth about these other functions, but if you go on to take genetics, you'll get a lot of that information. And if you're interested in that, that's great. If you're not interested in that, then whew, buckle up. All right, you should be interested in it because it's like really cool, but that's just me, somebody who loves genetics. Okay, so let's move on, shall we? At this point, you know how proteins or how genes lead to the creation of proteins, so how to go from a genotype to a phenotype. You know the details of that process and also how that process is regulated. Now, let's talk about what happens when there are mistakes in that process, which are quite common. So first off, just to kind of talk about what constitutes a genetic mutation. So remember, in this case, we are talking about a direct change in the nucleotide sequence of a cell's DNA. If we are changing the nucleotide sequence, we are changing the genotype. So just a reminder that this is different from epigenetic change. Epigenetic change, again, is when we don't change the genotype. So we're not changing specifically the nucleotide sequence, but we still may see an effect, as we'll talk about. We can still see all of these effects. Maybe not a frame shift mutation. Well, yeah, a frame shift mutation. But whenever we're talking about genetic mutations, what we're talking about is a change in the actual nucleotide sequence of a cell's DNA. So we can categorize these changes based on kind of like what type of change has occurred in the nucleotide sequence. And we categorize these into two kind of broad categories: point mutations and frame shift mutations. So anytime you hear the term point mutation, what you would think about is it's just like a change or a switch in one nucleotide. So we'll look at these specifically here in a minute. But point mutations, think about like if you have a strand of DNA and you just like point, just one nucleotide is affected. These are primarily via the mechanism of what are called base substitutions. So remember that base is referring to nucleotide. So if you talk about base substitutions, it means like one nucleotide is changed to a different one. So like A to C or something like that. So as we'll talk about, point mutations can be pretty drastic. They can cause a pretty large change, or they may not have any effect at all. So let's talk about those here in a minute. So point mutation affect one single base pair in the DNA. Alternatively, we can have what's called a frame shift mutation, and a frame shift mutation would affect the entire reading frame of a gene. So first off, we need to know what a reading frame is. So a reading frame would basically be the entire nucleotide sequence, right? So a reading frame of a gene would be from a promoter, the sequence from a promoter, from a, sorry, it's difficult to write or type and talk at the same time. The reading frame would be the entire sequence of a gene from a promoter to a terminator. So what a frame shift mutation could do is basically it would change if it occurs. Usually it's via two mechanisms, either inserting a nucleotide or removing a nucleotide. Either way, the fact that your nucleotide sequence is important in sets of 3. Remember, because every 3 nucleotides will lead to the code, eventually a codon, and it's those 3 nucleotide sequences, those codons, that code for amino acids. So if you are to remove or add a nucleotide, it messes up those trinucleotide or codon sequences. So a frame shift mutation would have much more drastic consequences because it affects the entire reading frame, so the codon sequence. Whereas the codon sequence, that's actually a really good way to say it, affects codon sequence, as opposed to like a point mutation, which may affects just one codon. So the key word there is that point mutations affect one codon because they're affecting just one nucleotide, whereas frame shift mutations affect a codon sequence, so a sequence of nucleotides. So we're going to look at this here in a minute. Okay, so before we get started on that, let's first kind of talk about point mutations more specifically. So remember that point mutations affect just one single base or one single nucleotide in a DNA sequence. So for example, you can see up here, what this picture is showing is a substitution. You can see if this is our normal gene, right? Technically, this would be our normal RNA. So it is really important here to note that genetic mutations typically take place in the DNA. So the change that occurs that we see in the mRNA would have taken place in the DNA, but it's just like seen in the mRNA, if that makes any sense. So we have our normal gene, which was transcribed, or our normal mRNA that was transcribed from our normal gene. And you can see that, again, 3 nucleotides at a time are a codon, and those codons code for amino acids. So our normal gene had codons that coded for this protein sequence: met, methionine, lysine, phenylalanine, glycine, alanine, right? But what happens is a genetic mutation in the DNA, which caused us to have an A in this codon instead of a G, which is the correct nucleotide. What happened is that substitution of nucleotides changed the codon, and that ended up changing the resulting amino acid. So remember that amino acid sequence is our primary protein structure. So if you are changing the amino acid sequence, you're changing protein structure, and since structure equals function, that could potentially change protein function. So that would change your phenotype. Whether that means something or not, we'll talk about. Okay, so this is just showing one type of point mutations. Again, this is an example of a base substitution, but we can actually classify base substitutions based on their effect. So how are they affecting the amino acid sequence? So we have a couple possibilities, right? So one, you could have this substitution change that nucleotide, and if that change in the nucleotide doesn't do anything, right, then like you would not have a change in amino acid. So what the heck am I trying to say here? Let me see. I hope did I not? So for this to make sense, I guess what I'll say first is that would be a silent mutation. If we had a substitution, a nucleotide was exchanged for another, and this did not change the amino acid sequence in our protein, that would be a silent mutation. Silent because it's not noticeable. So how would this happen? And in order to understand that, we need to remember that the codon table is repetitive. So for example, let's take leucine here, this amino acid. Let's say that our initial codon was UUA, but we had a genetic mutation and the nucleotide substitution was a G for this A. We go from our sequence being UUA to UUG, but that still codes for leucine. So because it still codes for the same amino acid, that change is basically unnoticeable. It is a change in genotype, but there is no change in phenotype. Okay, that's a really good thing to say. Let's do that. All right, there is a change in genotype, but no change in phenotype. Okay, right, so again, the nucleotide did change, so your genotype did change, but because of the fact that the codon table has that repetitive nature, what they call degenerate, which I will never understand for the life of me, but because the codon table is repetitive, you could have a mutation, and that mutation does not change the resulting amino acid. So again, that would be a silent mutation. Alternatively, what could happen is what was exemplified in the picture on this slide, which you have a genetic mutation, so again, a substitution of nucleotides, and that does lead to a change in one amino acid. So again, you can see here that what we have this codon originally is GGC, which codes for glycine, and that mutation causes the codon to change to AGC, but that change does change the codon, and that change does end up changing the resulting amino acid. So this may or may not impact protein function. And that goes into a little bit more about stuff that we didn't necessarily talk too much about. But if you remember from a while ago, when you did the Learn Smart and you talked about protein structure, and you talked about primary and secondary and tertiary and quaternary structure. So I know I'm reaching way back, but it's important stuff. So remember that protein folding and that really important protein structure, the 3D structure of a protein, depends upon chemical interactions between those amino acids. So what could happen, for example, is that even though we get this change from glycine to serine, it could be that serine and glycine are chemically similar enough that it doesn't affect how that protein is folded. So for example, if glycine and serine are both like relatively acidic, then their properties and their effect on protein folding may be so similar. So you do have a change in genotype, but that in that case, if the chemical properties of those amino acids are similar enough, that may not change phenotype. However, let's say that that nucleotide changes the amino acid to an amino acid that is chemically quite different. So for example, if glycine is an acidic amino acid and serine is like a basic amino acid, right? Polar opposites, that may impact protein folding, and therefore that would impact protein structure and protein function. So missense mutations, what they do is they do change the resulting amino acid, but the effect of that change completely depends on what amino acid it's changed to. So basically the comparison of the chemical properties between the changed amino acid. And then another possibility of a base substitution is what is called a nonsense mutation. So in order to understand a nonsense mutation, we need to revisit that codon table again, just to remember that within the sequence, or within all of our variety of codons that exist, there are 3 different codons that code for translation to stop. So this should be at the end of an mRNA sequence, obviously, but what can happen is, let's say that initially in your protein sequence, right in the middle of your mRNA, you had a codon that's UAU, and let's say you have a genetic mutation that changes that U to an A. So initially U coded for tyrosine, so you would just keep going. But if that mutation caused a U to an A and a stop, basically what would happen is translation would stop right there. And if that's in the middle of the mRNA, only half your protein is made. So that would be called, where'd it go? A nonsense mutation. And so again, that terminates translation early. So I have a question. A question is, so a missense mutation happens in mRNA. All of these mutations are occurring in the DNA, but the effect is in the mRNA as well. So for example, I know this picture is a little misleading because it's showing the mutation in the mRNA, but just note for any of these, where this change actually took place was in the DNA. It's just that our mRNA reflects that change because our mRNA was created via complementary base pairing. So I'll kind of elaborate on that a little bit later. I think what I'll do is I'll try to give you some examples of that. I'll get that prepared for Thursday because it's a little bit confusing the way this picture is showing it. All right, so again, yeah, all of those changes are taking place in the actual DNA, but the mRNA just reflects that change. So a silent mutation, again, is where you get a mutation, so your nucleotide is changed, but that doesn't change the resulting amino acid because of the fact that the codon table is repetitive. So you have a change in genotype, but not in phenotype. A missense mutation is where, again, shown in the picture, where you get a change in nucleotide, and that does change the resulting amino acid. The severity or the kind of result of this change, whether it truly impacts phenotype or not, is completely dependent on the chemical properties of the amino acid that it is changed to. And then a nonsense mutation is when we have a nucleotide exchanged, and that exchange leads to the amino acid being changed to a stop codon. So again, this would basically stop translation early, which we definitely don't want to do. So you can think about this, silent, it's not changing anything. That's why it's silent. It's secretive, it changed the genotype, but that doesn't matter, it's silent. Missense is an actual mistake, right? So you do see the wrong amino acid, right? A different amino acid. So it's a mistake, missense, mistake. And then nonsense, you have translation stopped because you reached a stop codon too early. So the change caused that amino acid to change to a stop codon. And so nonsense, no sense, no translation. It stops right there. So that's kind of how I would remember it. This picture is just showing these, again, sort of like visually. You have and this is just a picture taken directly from your textbook. So hopefully you also saw this in the Learn Smart. But you can see here, this is maybe a little bit more clear than this picture because your textbook does a good job, because it is showing the DNA. Good, all right. So here we have our normal correct structure of our gene, our correct gene sequence, ATG, GCC, GGC, etc. That would be transcribed into mRNA, and that mRNA would contain codons for the amino acid sequence, methionine, alanine, glycine, proline, lysine, glutamine, threonine, I think. All right, that would be our normal gene. If we had a silent mutation, this would mean that our amino acid sequence is not changed, so again, our phenotype is not changed, despite the fact that our genotype is changed, again, because of the repetitiveness of the codon table. So even though we see a change from CCG to CCC, those codons both code for proline. So no effect, it's silent. If we see a change, let's say in this codon, so our normal gene is ATG, GCC, and we change to ATG, we have a mutation where that G is changed to a C. CCC codes for proline, so we had a switch from alanine to proline, so our amino acid sequence is changed. And again, we don't know if that's going to be a problem or not, because it all depends on how similar or different alanine and proline are. And then finally, we see what happened here is we have this sequence was AAA, and it was changed to TAA. And what happened then is that changed this amino acid, or this codon, which originally coded for lysine, into a stop codon. So that would mean that translation would stop right there. So we have an early stop to translation, which is a nonsense, no sense mutation. So I had a question. How many cells with mutated DNA making the wrong proteins does it take to alter the phenotype? Or does just one mutated genome make a huge impact? That is such a good question, and we're going to talk about it. I have a whole slide dedicated to that question, so good job. Right, so I'll tell you, basically that's like a precursor to that. It completely depends on the cell. It completely depends on what cell the mutation is taking place in. So we'll get there. So those are all the different types of point mutations. Again, just making a change in a single nucleotide. However, we can have a little bit more severe consequences when we have what's called a frame shift mutation. So what I will say is, for the most part, where the effect of a point mutation can be relatively minimal, right, depending on if the missense results in a chemically different amino acid or not, or pretty major with a nonsense mutation stopping translation. Depending on where that stop takes place, it can be relatively severe, or it can have no effect at all with with any of these, really. So the severity of point mutations kind of varies. However, frame shift mutations, my mouth would not say that, I apologize. Frame shift mutations generally will have an effect. And that's because, again, instead of changing just one codon, they're affecting the entire codon sequence. So how do they do that? Here you're looking at frame shift mutations. Again, this is kind of referring back to that original picture, but I also have kind of a different picture here. If you have frame shift mutations, this occurs in two different ways. You can have insertions or deletions. So you can have a nucleotide kind of like inserted randomly, or you can have a nucleotide removed randomly. And what this would do, again, because instead of just substituting one nucleotide for another, which would mean just one codon is affected, you can see just like pulling out a nucleotide kind of makes all of these nucleotides shift left in the case of deletion, or shift right in the case of insertion. So instead of affecting just one amino acid, it affects the entire sequence, subsequent sequence of amino acids. So you can see with insertion, let's say originally we had AUG, AAG, UUU, GGC, GCA. We insert a G right here. And then now we have UUG, and notice that our second codon, instead of being GGC, is now UGG, and then this one is also affected. So all downstream nucleotides are affected. Same with deletion, it just kind of pushes this G into this spot where this U was deleted. So again, the effect of a frame shift mutation is that as opposed to just having like one amino acid changed, you have the entire subsequent reading frame changed. And you can also see that in this picture where this DNA sequence had an adenine, it looks like an adenine inserted, right? Yeah, an adenine inserted. And you can see that it has the entire amino acid sequence changed. So obviously a lot more drastic consequences if you're affecting way more amino acids than just one. It's much more likely to affect phenotype. In fact, I would say it's almost guaranteed to infect phenotype, I guess, depending on where it is. So I do want to mention that we have been talking really about what happens when these mutations occur in a coding sequence, because we're talking about phenotype, right? So this is what's going to happen to the phenotype if we affect the genotype. But that's really limiting us to just that 1.5% of our DNA that is exonic, right? Codes for proteins. But we can also have these mutations take place in non-coding sequences of a gene. For example, the promoter, or outside of the gene in regulatory sequences like the TATA box, or it could occur in like a splice site, right? So it could change how exons and introns are spliced. Or maybe outside of a gene, it could take place in an area that codes for a translational regulatory element, maybe something like what's called a spliceosome, which is something that like codes for something that does the splicing, or maybe something like that codes for maybe something that regulates like mRNA activation, right? Codes, I don't want to say iron, but because that would be a phenotype, but maybe something like that. Or something that's an intergenic region, so that would be like an intron. You can see that's not as likely to affect gene expression to our knowledge. So a couple of things that can happen here, right? If you have a mutation that's in a promoter, what can happen is that may affect the ability of RNA polymerase to bind to the promoter. So there's two things that could happen. This could actually increase or decrease the rate of transcription. Let's say you have a genetic mutation in the promoter for the gene that codes for insulin, and that makes you create less insulin, right? So that would be a deficiency in insulin, which is important for cellular respiration and energy. So it could increase or decrease the rate of transcription of certain genes. Same things if it occurs in regulatory sites, splice sites. Basically, you're affecting with your ability to regulate how often you are creating proteins. So I want to mention, I think this is probably where we're going to leave off because we're running out of time, but I do want to mention that of course there are disorders that are notable to human disease that result from genetic mutations. So I've kind of just listed here some of them, but I think this is actually a pretty good place to, I know we didn't fully finish this lecture just because it's cool, but I think what we'll do is we'll pop in here on Thursday and finish out this lecture, because I don't want to just rush through the diseases. This is important stuff. And I know probably stuff that you guys are actually interested in, so I don't want to gloss over it. So I think this is where we will stop and where we will pick up on Thursday. What day is it? With that, or if you have any questions though, so I had a question about roughly how many cells was mutated. So again, I'm gonna like copy that over because that's a really good question. I wanna look into that other question as well. We're also going to look into this question. And we're gonna look into this question as well. So I will have those answered for you on Thursday because we'll finally have got to the point where I can answer those questions. But if there's any other questions today, go ahead and ask them about what we covered today. I will still have office hours. Those office hours will be from 11:30 to 12:30. They may start a little late because I have a meeting that's going to bump into them. So if you need office hours, I think today probably the best thing to do would be to just email me and we can make sure you get those office hours. All right. So I think if there's no questions, I'm going to go ahead and stop sharing my screen. Maybe. There we go. But if there's any questions about any of the material, go ahead and type them in the chat box. Otherwise, have a wonderful, wonderful day. And I loved hanging out. And email me. All right. Thank you all. See you soon. See you Thursday.